All right, so what we're going to do today is go on to what is, in essence, the cutting edge of the latest view of the tree of life, or a 1.5 to 2-year-old view of the tree of life. And it's in some ways similar to what we've been leading into, and in some ways somewhat different. And then we're going to move in the next week into characterizing organisms directly from the environment. Up until now, most of the general concept for most of the work on the tree of life originally was working, of course, on organisms that you could grow in the lab. Some of the data that's in these papers comes from organisms from the environment, as discussed in particular in the second paper, the Williams et al. Paper. So these are the two papers. I really, when the second paper here, the Williams et al. Paper came out, I really didn't want to read it. It made me sad that there might not be 3 domains in the tree of life. And I confess I avoided reading it until maybe a month ago. I know Martin Embley really well. I've collaborated with him on and off for years. He's completely brilliant. But I avoided it mostly until very recently. The first paper is the culmination of a project that this professor at Smith College, Laura Katz, ran on what's called the eukaryotic tree of life. There was an NSF program on the tree of life, and she got a grant to sample the phylogenetic diversity of eukaryotes and gather more gene sequence data from this. And I love this paper. It's really well done. There's some technical details in there that even I skim over. So, but I just think it's just a really good summary of what we know about eukaryotic diversity. And I like that Laura is, Smith is not a research university. It's a liberal arts college, and most of the people involved in this have either been sort of undergraduates in her lab or graduate students that are jointly with her in other places. And a few years ago, I went to a conference that Laura was speaking at, and my wife and kids came, and it turns out Laura was in my wife's class in college. I didn't even know that. So my wife and Laura knew each other really well. So before we go into these papers, I thought I would spend a few minutes on a little phylogenetics. I sort of hinted at this before, but I had put together a couple of slides just to both review what a tree is and then review in particular some of the different ways different worries people have about phylogenetic trees, why they can go wrong, and different approaches to assessing the significance of the results. And please, any time along the way here, stop me if there are any questions. So this is sort of the summary of everything you need to know about a phylogenetic tree, probably more than you need to know. But a phylogenetic tree is basically showing the evolutionary history of a group of taxa. In some cases, when we call them taxa, we refer to species. In other cases, we might refer to genes. Phylogeny could be the evolutionary history of any object, whether it's species or phyla or individual genes. And basically, the way to read a phylogenetic tree is pretty simple, but you should practice this over and over and over if you want to read through these papers and sort of get them quickly. These are the termini, or the tips of the tree, and that's where you put the taxa. Each little circle that I've shown here is a node in the tree. A node represents a point in the tree at which there's some change in the branching in the tree. If we're at the end, when the branch ends, that is in modern times, they're called terminal branches that go to terminal nodes. And then any of the other nodes are called internal nodes, and any of the other branches are called internal branches. And basically what you're trying to represent here is the you can view this in two directions, basically. If you think of a tree going from the present to the past, what you're representing in a tree is the common ancestry of the taxa, or the objects at the tips. And so if you start at the tips of this, we see that G and H share a common ancestor here at this internal node. And then you march your way backwards into the tree to the point where F, G, and H share a common ancestor, and go further back to the point where E, F, G, and H share a common ancestor. And so in essence, you can view a tree as representing the common ancestry and the patterns of that common ancestry among the organisms or genes that you're drawing. The tree also can be viewed as going from the past to the present and thinking of splitting events in the history of species or genes. That is, you can go from one organism or species in the past, and then simplest is to think of this in terms of speciation and species of history. You have one population in the past that gets split into two descendant populations. You represent that by drawing a node and two branches coming out of that node. Now, when this initially happens, the two descendant populations are very, very similar to each other. You can imagine a river coming in between, in the middle of a group of organisms that can't swim. So on the left side of the river and the right side of the river, they're basically two populations that were the same species. But over time, they're going to accumulate differences until they become distinct species. And more and more time, they become more and more different. And so we represent that by just drawing branches in the tree. And I think it was you who asked a question, or someone asked a question about oversimplification in trees before. We don't really try to represent everything that's going on within a species or within a population when we draw these trees. So if this was primate evolution, in the past there could have been a population of the common ancestor of humans, chimps, and gorillas that could have been 50,000 individual primates of some kind. They would have a genetic diversity among them. They would have, males and females. They would have differences in different regions of the world. We're not representing any of that. We just draw a spot. And then if there was some speciation event such that one group became, the common ancestor of humans and chimps, and the other group became the common ancestor of gorillas, we're just going to represent that by two different branches here. And eventually, maybe there are other speciation events, so this could be humans, chimps, and gorillas, and so on. And we're glossing over a lot of the details when you draw a phylogenetic tree. There's, unless we had data from every single individual in a population, which eventually we will have for some organisms, you can't really represent that total diversity. So the last part that's really important, and we'll get into a little detail on this, is that as you go way back into the past, you reach a point where all of the organisms in your tree share a common ancestor at that point. And this is called the root node of the tree, the most recent common ancestor of all the organisms in the tree. And then this branch leading up to that root node is known as the root branch or root of the tree. And basically, this represents evolutionary history that occurred prior to the existence of the common ancestor of the organisms, the most recent common ancestor of the organisms you're looking at. Those sort of terms basically make sense. I'm not going to show this, but tree can be rotated. This is on a piece of paper. You can rotate it 90 degrees. It means the same thing. Some of the papers we'll see will have trees going in all sorts of different directions. The point is to figure out where the past is and where the present is. And all the trees mean the same thing once you do that. So for inferring phylogenetic relatedness, I already sort of mentioned this, but I just want to go over it again. What we want to do is find heritable features of organisms to study. These are known as characters, or character traits or traits, and you will see each of those terms get used in many of these papers. And the reason you want heritable features is what we want to do is compare organisms to each other, and hopefully the features that we're comparing have some connection to the history of those organisms. So you want stuff to be heritable in order to reflect history. And the particular form that one of these characters takes is known as the character state, or sometimes just the state. And there's millions of different types of characters that people have used for inferring phylogenetic trees. Almost everything we will focus on in this course, although there's some discussion of others in every paper we'll probably look at, but we're going to focus on what are called molecular characters, and largely sequence characters. So in this case, the character that we're looking at is going to be either a gene, and then the character state for that gene will be present or absent. Or we will zoom inside those genes and look at individual bases in the DNA or amino acids in the protein. And then each of the positions in the gene will be a character, and the particular nucleotide or amino acid used will be the character state. And the way we represent this for any type of data is what's called a data matrix. You have taxa in rows and characters in columns, and the character states are what you fill out inside the boxes. For sequence data, this is what we do is we represent this as an alignment. A sequence alignment is the data matrix of molecular characters. And the rows correspond to either individual genes or individual species, and the columns correspond to positions within those genes. And then you fill in the box with the nucleotide or the amino acid, depending on whether you're looking at gene, DNA, or protein. Does that make sense? We're not going to go into a lot of detail on alignments, but lining up the different genes from different organisms is an incredibly critical step in doing phylogenetic reconstruction. And to think about this, I find it useful to think about evolutionary history starting from the past to the present and imagining how genes evolve. And so if you think of a gene in the past, let's just make it simple. Let's say its sequence was AAAA, 6 A's. And then there's a divergence event. Initially, after this divergent event, the two descendants are basically could be identical to the parent. So this is a little bit confusing to people. So this is why it's easier to imagine to not think about the fine scale details of exactly what's going on at these split events. So a split could be used to represent when two populations split into two different parts of the world. But initially, they might be identical in their sequences. And over time, eventually, they're going to accumulate differences between them. And so let's say this sequence deletes one of the A's. And now it has 5 A's in its sequence. And let's say this sequence changes one of the A's to a T. And they continue to evolve. Maybe this sequence adds 2 C's in front of the T. And this sequence changes one of the A's to a C, and so on. And what we want to do with a sequence alignment is build a matrix such that when we have our modern data, we want to make sure that we line up the specific positions that have a common evolutionary history of that position. So this A came from that A. This A came from that A. This A changed to a T. And so on, and we line them up. And we line up these. There's a problem here because we lost one. Let's pretend like we know that the third one was lost, and so on. And we want to line them up. And that's what this sequence alignment represents. is the lining up of particular individual positions in the different sequences that share a common history. Really important to do that. It is very, very hard when a lot of evolution has happened to make one of these alignments that works well. Sort of the equivalent of trying to figure out, when you have, if you just had a human and a snake, and you were trying to figure out which bones corresponded to which bones between humans and snakes, you'd have a panic attack and probably not want to keep doing it. But eventually, if you had a lot more data, you could line up humans with the diversity of mammals and then compare the diversity of mammals to the diversity of birds and reptiles and eventually figure out that some bones corresponded to certain other bones between these taxa. And we do the same thing with sequences. We try and figure out ways to line things up even when they might be reasonably different. That's what a sequence alignment is. Fortunately, for most of the papers we're looking at, ribosomal RNA is reasonably easy to line up between organisms. Some other genes are not. That the general concept here basically makes sense, what you're trying to do? We're going to glaze over it. They talk about that in these papers, and in every other paper we will probably see there will be some discussion of how they made their sequence alignments, how they tested their sequence alignments, how they threw out some of the data that they didn't trust and kept some of the data that they did trust. So once you do that, once you have one of these alignments, there's 5 major classes of methods that you could use to build an evolutionary tree from that data. Each of those classes have from 5 to 1,000 different sub-methods to use within those data. I'm going to focus on the 4 commonly used classes of these approaches. So there are 4 sort of reasonably commonly used methods. Depending on who you are, which method you use, the frequency with which you use them is going to differ. So the way it works is pretty straightforward. You take a sequence alignment, and for the parsimony, maximum likelihood, and Bayesian methods, what you do is you draw possible trees to relate the taxa that you're looking at. So if you had humans, chimp, gorilla, mouse, and fish, or something like that, you have 5 taxa, you would start to draw all the possible ways you could draw evolutionary trees to relate those 5 taxa. So in one of the trees, humans and chimps could be together, and then mouse and fish, and you could test what it would mean if gorilla was the outgroup to the rest of them. And then you're going to draw the next possible tree, and the next possible tree. And you draw all possible trees that relate the organisms that you want to study. As you get more and more taxa, this increases more than exponentially, so you can very rapidly have more trees than molecules in the universe. So people don't actually test all trees. There are algorithms that scan through possible trees and try and find different trees. And what you do is you take tree number 1 and you give it a score. Now, parsimony methods give it the score based upon one approach. Likelihood gives it a score based upon another approach. Bayesian gives it a score based upon yet another approach. But the concept is the same. You take tree 1 and you give it a score. You take tree 2 and you give it a score. You take tree 3 and give it a score, and so on, until you've tested as many trees as you think is sufficient to test. And you pick the tree that has the best score as the winning tree. And that tree is the one you say best represents the evolutionary history of your taxa given your data. Parsimony methods, what they try to do is count the number of evolutionary events in the history of your data, and they try and minimize the total number of evolutionary events. Likelihood methods apply a statistical model to the data and try and find the tree that has the highest probability of generating the data you observe. It's a statistical model. Bayesian methods are very similar to likelihood methods, but they use a different type of statistical approach where you're actually trying to calculate the probability of the data and the tree itself together. but they're the same concept. And all three of them are basically the same idea. You test different trees and you give them scores and you pick the best one. Distance methods are fundamentally completely different. That's the one I diagrammed in the last class. Distance methods, you use an explicit mathematical formula to take the data and calculate a tree that represents the data. You don't scan through different trees. You have a formula that you apply to the data itself and algorithmically calculate the winning tree. That's what we're going to do for phylogenetic methods here, unless you have specific questions. And of course, along the way, as we get into other examples, I'm happy to talk about these. We work on developing these methods in part in my lab. But for the purposes of this course, all we really need to know is that there are these different classes of methods that people disagree on which is the best method to use for which particular data set. And what they do with that disagreement is either, as they did in the Laura Katz paper, they use multiple tree building methods and see what it says about their data. Or they pick their favorite method and cite a bunch of papers that say that that's the best method to use, and then they just use that method. And there's lots of examples we will see of both approaches. Now, my personal opinion is that maximum likelihood methods are the best methods to use. Bayesian methods, I think, are conceptually superior to maximum likelihood methods, but the software for doing them is still in development because Bayesians were the most recent methods that were developed. So in my lab, if we can choose, we probably end up using maximum likelihood methods whenever we can. But they all have their pluses and minuses. Yeah?
For the distance method, is this mathematical formula, is it like a general formula applied to any tree, or does it differ based on the tree you're trying to use?
There are hundreds of these formulas, and which one you use depends in part on the type of data you're using, and you can adapt which one you use based upon the general structure of the tree. If you have, for example, taxa that are really closely related, you might use one distance approach versus if you have taxa that are very distant. And the main reason for that is these distance methods, what they try and do is first calculate a score between each set of taxa that represents not how similar they are, but how evolutionary distant they are. And as you get further and further apart, you need to do corrections for that evolutionary distance because you can't quite calculate. It's not as easy to calculate as things become more and more different. And then the tree building method that you would use with that data, the best ones vary as you get further and further apart. So there's, literally hundreds of different approaches to do this. I have my favorite distance methods, as do other people if they use distance methods. A lot of people use a method called neighbor joining. And the main reason for that is there was really good software to do it, and it's very, very quick. So you can build a neighbor joining tree of 1 million taxa in a short period of time. You'll run it on a computer cluster for 6 months to do a likelihood tree with 1,000 taxa. So distance methods are really good for massive data sets, although they have flaws. And I'd be happy to point you to the literature on this and discuss the different bells and whistles, but it's not going to matter that much for most of what we care about here. So there are a few things that it's useful to know, even if we haven't picked our favorite method. There are lots of things you can do about trees that you can do to basically with all methods. So one thing you should think about, and this is in both of these papers and will be in many of the other things we talk about, is something called long branch attraction, LBA. And what this is basically, if you imagine, so I didn't say one thing, and I hinted at this in response to your question last week, but evolutionary trees have two different ways that they draw the data. You can draw the data such that all you're trying to show is the relationships among organisms. And that's called a cladogram, clade being groupings of organisms. You can also draw a tree such that the length of the branch in the tree represents the amount of evolution that has happened in that lineage. So organisms that evolve more rapidly get a longer branch than organisms that evolve slowly. So HIV, for example, would be at the end of an incredibly long branch compared to humans, which evolve much more slowly. And so what you do in these cases is you change the length of the branches. But it's important, if you saw a tree like this, what this basically means is that A and B share a common ancestor here. They've been evolving, let's say these are modern organisms. They've been evolving for the same amount of time since this was a common ancestor in the past. With a longer branch over the same amount of time, that means that this one has evolved more rapidly. So let's say its sequences change more rapidly, or if you're doing a morphology-based tree, its phenotype changes more rapidly. So what this tree on the top here shows is a hypothetical model of the evolution of 5 taxa. I didn't draw it, but here's the root down at the bottom. And what this model basically shows is that first you have lineage 5 splitting off from the common ancestor of everything else. 1, 2, 3, and 4 have a common ancestor here. And then there are 2 groups. 1 and 2 are together, and 3 and 4 are together. But on the branch after 1 and 2 separated, 2 started to evolve at a higher rate than 1. And on the branch after the 3 and 4 separated, 3 started to evolve at a rate higher than 4. But 3 shares a common ancestor with 4 to the exclusion of everything else. And 1 shares a common ancestor with 2 to the exclusion of everything else. Unfortunately, most phylogenetic reconstruction methods, all of the ones that I showed you before, have a flaw, and that they are prone, if you took data from these organisms with this history and you fed them into a phylogenetic reconstruction method, those methods would want to group 2 and 3 together. Organisms that are at the end of long branches artificially get grouped together in most phylogenetic reconstruction methods. That's what's called long branch attraction. Now, some methods are more prone to this than others, but every method has this problem.
Is there any way to correct it?
So as in these papers, in particular in the Laura Katz paper, but also in the other paper, they talk about the general way to try to correct for this. There are 2 things. You try and throw away the data that has uneven rates of evolution. So it may be that if you had whole genomes from organisms, maybe not all the genes may not be prone to this. So you might want to try and find out which genes are evolving at uneven rates and throw those away. And the other thing you try and do is fill in the branches. So if you saw a pattern like this and you were worried that 2 and 3 were not in fact closely related to each other, what you would try to do is find other organisms that would split up these long branches. So a sister taxa to 3 that was more closely related to 3 than it was to 2, you could fill in, and it would start to break up the length of this branch. And that helps solve this long branch attraction problem. So taxon sampling, which is a big issue in both of these papers, is really important in particular for breaking up this issue of long branch attraction. That sort of makes sense? So this is related to an issue, basically due to an issue called homoplasy. So homoplasy is basically convergent evolution of the character states for the characters that you are looking at when you're studying organisms. So the reason that long branch attraction is basically a problem is that with this extra evolution that happens on the branch leading up to 2, and the extra evolution that happens on the branch leading up to 3, they may accumulate changes in regions of their gene or their genome or their morphology that actually make them artificially look similar to each other for that, just because there's lots of change going on. And this is also known as homoplasy. And you'll see this, we don't need to go into a lot of the details of it, but you'll see this in many of the papers. So identifying homoplasia and correcting for long branch attraction are very similar issues. So then there's two things that I just want to quickly mention, although the first one that's shown here is much more commonly used. There are many statistical approaches that people use to try and figure out if their tree accurately represents the data that they have. Now, these methods aren't testing whether or not the tree is accurate. They're simply saying, I have a bunch of data. And let's say the best way to think about this is, let's say I use a distance method. And I have the data, and it actually calculates a tree. And it draws you a tree. Which parts of the tree are robust? Which parts of the tree represent all of the data, and which parts of the tree might be, representing only a tiny portion of the data? And one way to test for this is something called bootstrapping, which is mentioned in most phylogenetic papers. In essence, you're taking your sequence alignment and you're resampling it, and you're generating a new alignment where some of your alignment columns are represented twice, and some are represented not at all. And you redo trees now with the new sequence alignments. And then you ask, how many times do I see this particular grouping in my new data set, where I've oversampled some of the data and undersampled some of the data? And if you see the pattern every time you do bootstrapping, that tells you that that pattern is very robust in your data. That is, all the different parts of your data show that pattern. So a common statistical method is not just used for sequencing, but it is the standard for testing how well your tree is representing the data that you have. There's another one called jackknifing. You will see this in a couple of papers. It's not as commonly used. It's resampling the data, but without making, having one be represented multiple times. You're just getting rid of some of the data in essence. So a second one that is much more discussed in these papers, and as we get more and more genome data is very common, is something called congruence. What congruence means is you take tree number 1 that represents the evolutionary history of a set of taxa, and you take tree number 2 that represents the evolutionary history of a set of taxa, and you ask, how similar are the branching patterns between those two trees? How congruent are the topologies of those trees? And you can ask yes, no, are they identical? That is, are they congruent? Or you can measure how different they are. You can calculate a distance between the trees that measures how different they are from each other. And then the last issue, which if we have time, we'll get into again at the end, is this issue of rooting trees. And the main reason that this is an issue is something I hinted at before, but I'm just going to go over for a minute, is that if you have 4 organisms, and data from those 4 organisms, and you want to draw an evolutionary tree that shows you how related they are to each other, the methods that we use to infer evolutionary trees can't figure out where the root is in the tree. And what they do is infer what are called unrooted trees. So they're basically trees such that, so if we had a tree with a root, imagine just erasing the root branch in the tree and erasing the root node and straightening out this branch here. And now drawing a tree where there is no direction to time in this tree. There's no past to the present. There's just branching relationships among organisms. This is sort of a mathematical trick where one branch is missing. And the problem with this is, if you have 4 taxa, for example, what we do with phylogenetic methods is we ask which of the trees is best for those 4 taxa. And it turns out with 4 taxa, there are only 3 possible versions of this unrooted tree to draw. You can have A and B together and C and D together, or you can have A and C together and B and D together, or you can have A and D together and B and C together. And the problem with any of these, let's say you do a phylogenetic analysis with parsimony, likelihood, Bayesian, and distance methods, and they all tell you that this tree is correct. The problem is we're still missing that root branch. And for this tree, there are 5 places we can place the root branch that all give us this same unrooted tree. And if we untangle these, they give very different stories about the evolutionary history of organisms. So if this is our root branch, the resulting tree will have B branching off first, and then A branching off next, and C and D together. And if this was the correct tree, you would have A branching off first, B here, and C and D together. And so these tell very different stories. Imagine this was archaea, bacteria, and eukaryotes, about the order of branching among these groups. So the ribosomal RNA tree that Woese built was an unrooted tree. He just, as I mentioned, he sort of punted on the issue of where to place the root in that tree. And we'll come back to that. And then the last thing, before we get into the papers, is this issue of masking. This is figuring out which parts of your data set to throw away before you build the tree. Usually what people do is they statistically analyze a sequence alignment, and they say, well, some of these columns in the alignment seem to be robust, and some of them are ambiguous, and we're going to throw them away. Oh, sorry. The last issue is then what people now are doing to combine data from different genes is to stitch them together into what's called a concatenation. So imagine you had 4 different genes from 4 taxa. Taxa 1, 2, 3, and 4. This is gene 1, gene 2, gene 3, and gene 4. You can just stitch them all together into one mega alignment and infer an evolutionary tree from this giant alignment as opposed to from the individual alignments. It's called concatenation. As we get more and more genome data, that's becoming more and more common. Yeah?
Why would that be more useful than, I guess, creating a bunch of trees and then congruently comparing them to each other?
Well, they talk about that in some of the papers. The main reason is that if there is subtle signal for the actual evolutionary history in your different genes, but it is very weak signal, it won't show up in an evolutionary tree of that individual gene. It will show up as an ambiguity. And if you sum up a lot of weak signal, if it's all pointing in the same direction, you get a stronger result. The problem is that if you stitch together things that have conflicting signal, then you are summing up stuff. You're making an average of things that don't exist. It's like taking the average height of, giraffes and mice and saying the average mammal is in between the two, right? I mean, it's averages of things that are different, in this case, don't mean anything. So these 2 papers are all about how can we figure out how to concatenate the data such that we are increasing signal and not increasing conflict.
Signal you mean?
I mean increasing your ability to resolve the evolutionary history of the organisms. So signal in the sequence data that accurately represents the history of those organisms. And as they mention in both of these papers, old events have a lot of conflicting information about that, and you might have signal, but it might be very slightly above the noise. And you might want to sum up many different pieces of that signal, just like, people do this all the time with images now. If you have 2 fuzzy images and they're of the same thing, you can overlay them onto each other, and a computer can make them higher resolution by combining the 2 images. That's the same thing you're doing here. But if you took, Justin Bieber and Lady Gaga and overlaid them onto each other, that's not increasing your signal. And that's the problem here, is that you're using the same type of statistical approaches to find weak signal in data. but you want to make sure that those gene 1 and gene 2, this only works if they have the same history. And we don't know that, and that's what they're testing in these papers. So again, here's the first paper, again by Laura Katz and Laura Parfrey, who was, I think, a postdoc in Laura Katz's lab, and a collection of other people that were collaborators with Laura on this project. The abstract is long, but does, again, I want to talk about how you read papers. What I do for this paper, when I first saw this paper, I scan through the abstract and try and figure out if it has anything in there that I will be interested in. And this certainly seemed interesting because it was basically saying we have started to resolve the phylogenetic relationships among the eukaryotic groups. So this is not the bacterium archaea, but this is really important both for the methods used in the next paper and because you need to know which eukaryotes to use to represent the diversity of eukaryotes in order to then resolve the evolutionary history of the supposed 3 domains. So I basically went through this paper and highlighted the sections that I thought were useful to me. So in the introduction, she's talking about these problems with phylogeny that I just sort of hinted at. The early events in eukaryotes happened a long time ago, like more than 1 billion years ago. Within each eukaryotic genome, the genes don't necessarily all have the same history. There's duplication events, there's deletion events. As we mentioned, there's gene transfer events, there's convergent evolution of sequences, there's rearrangements, there's all sorts of weird stuff. They just call this complex gene histories. There's uneven rates of evolution that now create long branch attraction in some cases and complexities. And, it's a mess. Oh my God, what are we going to do, basically? They're trying to tell you, we all do this. They're trying to tell you how smart they are. This is really hard. Look at how hard this is. And they discuss this big issue, which was the whole point of their project, which was, do you want to increase the number of genes that you look at? So if you're looking at 5 organisms, one thing you could do with money is, rather than look at just the ribosomal RNA, you could look at 40 genes in those organisms or 100 genes in those organisms. And another thing you could do to increase your sampling is to sample more taxa. And as they refer to, there's a long-standing debate about the relative benefits of increasing gene representation within taxa or increasing the number of taxa you sample. And there are so many papers about people testing this with simulations and with real data and with theoretical arguments. And long story short, they say, screw this, do both. Get more data from each taxon. The way they do this is random gene sequencing of the expressed genes in these organisms. And get more taxa. And therefore, you solve both problems. They do talk about which one they think is more important, but yeah.
I think it was one, but where they talk about using taxa that don't really have as much information as well. I don't know how that would help.
So yeah, this is a really important part of the debate about more taxa versus more genes. So what ends up happening when you generate a lot of data for new organisms is occasionally one of the organisms that you think is really important for your phylogenetic study, you have very little data from that organism. And so if your choice is, should I build an evolutionary tree with 100 genes, but if I say I need 100 genes, I only have 20 taxa represented? Or should I build an evolutionary tree with many more taxa, but if I do that, I can only do 20 genes across all those taxa? So this is their issue, is that there are some taxa that are really interesting or important that they don't have a lot of data from. And they basically do both. So they build a tree, as we'll get to, they build a tree with as many taxa as they could do, but with much less data. And they also build a tree with lots of data, but fewer taxa. And then they try and compare the results between them.
So is that why wild type organisms get ignored more?
Why non-wild type or?
No, wild type get ignored.
I don't know.
I mean, in other words, this thing, they're culturing all this in the lab, right?
but wild type is, for microbes, is the one that you cultured.
Oh, I see.
A wild type is basically the first one, or the type strain is
I
the
see.
first one that was cultured for a particular group.
Right.
So I mean, I've seen papers where they basically built a tree with each set. So they said gene number 1 is across 40 organisms. And if I combine gene number 1 with gene number 2, I'm now limited to 30 organisms. And if I combine gene number 2 with gene number 3, I'm limited to 20, and they do every possible set. What they do in this paper is just basically took three different versions of the sets. One with lots of taxa but fewer genes, one with lots of genes but fewer taxa, and one in between. Is that basically what you were?
Yeah.
And they go through in the introduction, reasons to do this include long branch attraction, taxonomic sampling issues, et cetera. What they did, again, was basically identify lineages that prior phylogenetic results said were evolutionarily novel, either based upon morphological features, which is actually what they use, this classification system of this guy, Paddy Patterson, who does morphological-based classification. And then they basically marched their way through all the different eukaryotic taxa and generated new genome data, and then scanned through that data to find sets that they could identify as homologous, made alignments for them, and then built trees. So they basically did in essence, three trees. They did a ribosomal RNA tree. That's the standard for eukaryotes too. And then they did alignments of these other protein coding genes where they concatenated them together and or added the ribosomal RNA to that concatenation and built trees with that. And then they used a couple of these methods to test for whether or not there was lateral gene transfer, basically to look for discordance, the opposite of congruence between different genes. And then they punted on the question of which method was best. They just said, let's use multiple phylogenetic methods and see what the similarity and differences are among them. And what they report is basically summarized in these two sentences. Whatever method they used and the different data sets they used, they recovered basically the same major eukaryotic groups. And the support for these, the statistical support, in particular with bootstrapping, was strong when they did not have a lot of missing data. As they sampled more taxa but had less data, the statistical support for their result went down. And then the rest of the paper is basically discussing and showing you these results.
So you're saying when so because they needed stable genes in order to accurately compare taxa, the number of stable genes that they could use went down as they increased the...
Yeah, it's not really stable genes. It's just that they were randomly generating data from these taxa, and they didn't always hit all the genes.
Okay.
So some taxa were just missing some of the genes. Their data was missing some of the genes' values. So then they show you, this is their tree with the most taxa that they could represent, that they felt comfortable with. I think most of these had all 16 of the genes. And doesn't really matter. I mean, if you care about eukaryotic evolution and the details, you might want to know the details of these results. That's not obsessively important here. They then zoom in on individual groups within their data. And by, this is what, they could find 16 genes across these 451 taxa that was eukaryotic wide. But then they said, I'll come back to those other things in a second. What happens if I just zoom in on one of those lineages? So this is just one of the 5 major groups of eukaryotes, and this is another one of the 5 major groups of eukaryotes. Now they have fewer genes across all of these, but it allows them to look at phylogeny within these groups. Whereas the point of this tree is really what are the relationships among the major groups of eukaryotes. And here is the data. So these are the figure 1 and figure 2 are lots of taxa, fewer genes, fewer taxa, more genes. And you can, see the similarities and differences between the results here. They punt on the eukaryotic root in this. So they're basically saying, we're going to look at the evolution of eukaryotes, and we really don't want to deal with the question of where the root is of eukaryotes. They show in the trees and also in this table some of the statistical support for the different patterns that they find. This is common in evolutionary studies. So usually presented as supplemental material in many papers now. But what this basically shows is the statistical support for some of these different patterns for different trees for the different groups. Bootstrap values go from 0 to 100. They're basically the percentage of times that you see a particular group in the different replicates that you make of the sequence alignment. Obviously, higher numbers are better. If you see the same group 100% of the time, that's a good indication that it's seen in all the data. Yeah.
So the RAXML that they use to get the bootstrap values, is that actually only for creating the bootstrap values, or does it also create trees?
RAXML is a program that takes sequence alignments and infers maximum likelihood. That's what the ML stands for, phylogenetic trees.
Okay.
And what you do is you basically feed it. It does it for you. It resamples the sequence alignment and generates multiple trees and then tells you what is the bootstrap value for different patterns.
So it's tree creation software.
Yeah. Used in both of these papers, actually, and commonly used. It's one of the better ones. The statistical, this paper I don't want to focus on that much. I want to get to the next paper, but I'm just sort of trying to take you through what people do. And again, they then zoomed in on the rhizaria, which is this group right up here. And then they zoomed in on the excavates, which is this group here. And then they basically tried to represent all of their data and results and the statistical support that they saw in this one figure. And this is the one that I find most useful. They've basically, they find these four major groups, 1, 2, 3, 4. And then for many other organisms, they can't figure out where they sit relative to each other. And what they do is they draw what's called a polytomy. What this means here is basically we have no idea where green algae fit relative to haplotypes or red algae or cryptomonads or this lineage. This means that they think that green algae are in this big group here, but they don't know where they sit. And so they're trying to tell you with this one figure what the good results were in all of their analyses. And so it's still a lot of ambiguity here, right? There are lots of groups, they can't figure out where they are. Within these groups, there's lots of polytomies, but it's still a pretty useful result for the people who study eukaryotic evolution to see what it looks like in these really early evolutionary events. They argue that the results that they're getting support the idea that taxon sampling is important, and they've collapsed what previously were 70 major eukaryotic lineages into what they call 16 groups. And that's the paper. And there's lots of detail I skimmed over. For example, if you read the whole discussion where they're talking about the photosynthetic organisms and they're trying to figure out what to do with. So photosynthetic eukaryotes are just a god-awful mess. And the reason that they're a mess is that the chloroplast that carries out photosynthesis, which originally was a free-living cyanobacteria, is so functionally important that eukaryotes are stealing it all the time from other eukaryotes. They're engaging in symbioses with other eukaryotes that have these chloroplasts. And when they do this, they bring in not only the DNA for the chloroplast, but the DNA for whatever the host was previously for that other organism that had a chloroplast. And then that DNA gets mixed together in the new organism. And figuring out, like, evolutionary history of a cell when the cell actually has 6 different evolutionary histories is a big mess. So they kind of tried to avoid many of the photosynthetic lineages or avoid a lot of the data from the photosynthetic lineages to not deal with this issue. Yeah.
Is that the same that's horizontal transfer, right? So actually
Yeah.
bring that up in the
Yeah.
introduction.
So what happens in evolutionary history, in particular with important functional compartments of cells, and the chloroplast is the best example of this. You have inside an algae, you have this photosynthetic machine. And many other organisms would like to make use of that photosynthetic machine. And sometimes they make use through just a sort of simple symbiosis. Turns out many times in evolutionary history, they bring in to their own cell another organism that does this photosynthesis. And then the genomes start to mix together and become hard to deal with.
Is that the background static, or is it you're trying to get a signal?
You're trying to get some type of history out of the data that you're observing. We would like to know what those events are in the history of an organism bringing a symbiont inside of it and then bringing another symbiont inside of it. And there are many people who study that. They didn't want to deal with it. They wanted to study the early events in eukaryotic evolution before even the chloroplast originated.
But you have to suppose that all the way along there's lateral gene transfer.
You do, and that's why they're testing for congruence. They're trying to find genes within each of these genomes that have the same history, and then sum up by concatenation the genes that have the same history and throw away the genes that have different histories. And the second paper does that much more explicitly. So this paper has a much even harder problem. They're going back even further into evolution. So if there is some amount of lateral gene transfer over time, as you go back further and further, it further obscures the signal of evolutionary history of organisms. And so they spend even more time in their paper trying to figure out what genes within these genomes have the same history. And again, the abstract does a pretty good job of saying what they're doing here. What they're doing is looking at the evolutionary history of bacteria, archaea, and eukaryotes. And just like the previous paper, they argue that there was not a lot of data in the previous studies. There wasn't good taxon sampling in particular. They're going to increase the taxon sampling by covering more archaeal diversity. That's the main thing they did in this paper. The introduction talks about the same types of issues. You go back further in time. It's a mess. They basically say there's this rooted 3-domain tree that I've talked about where archaea are the sister group to eukaryotes, and they want to test that. There are many competing papers that have been published, but the genome sampling wasn't good. So let's see what happens when we increase the number of the archaeal diversity that's represented. In particular, there are these 4 lineages of archaea that they have more genomic data from that they're going to include in this study.
When you say not good, how not?
The representation of diversity. So we will get to later in the course when we talk about genomics that I coordinated the largest project to cover phylogenetic diversity of bacteria and archaea. Which would even do better than this. But before people included these lineages, I would say you probably had less than 1% of archaeal diversity represented in the data they were using. Probably a 1/10 of a percent. And this makes it 5%. So it increased more than an order of magnitude of the total breadth of archaea. They still didn't sample it well, but a massive increase. It's sort of the equivalent of if you tried to build a eukaryotic tree and you had just yeast, humans, and Arabidopsis, and none of the protists, and that's the equivalent of what was happening with the archaea. Yeah.
Are you among a group or a tendency to develop a field guide for microbes?
Yeah, that's my idea actually. So yeah, I was the one who came up with the idea of doing a field guide to the microbes. We'll talk about that next week. That's the whole point of environmental characterizing microbes. That's my dream. But this is more about the deep evolutionary history. We're not yet into the field. And again, they go through, should we concatenate data? Should we not concatenate data? If the data is conflicting signal, what do we do? What happens with long branch attraction? And then they say, taxon sampling should be good. And so, if you look at they merge together the results in the discussion. I like doing this in papers. Some people like having results separated from discussion. They basically built a mega alignment where they combined together the 16S ribosomal RNA and the 23S ribosomal RNA into one ribosomal RNA concatenated alignment, and built a tree with 36 species total, bacteria, archaea, and eukaryotes. And then looked at the phylogeny. They determined that these two genes, the large subunit and the small subunit, appeared to be congruent, so they were comfortable with the concatenation of them together into a single alignment. And then what they're doing is not actually looking at the really early events in evolution. What they're saying is, we think that the root of the evolutionary tree, the universal tree, is either inside the bacteria or on the branch leading up to the bacteria. So what we're going to focus on is what happened after that, which is the eukaryotes and the archaea. And then they found in their results, which is shown in figure 1, something that is not consistent with the 3-domain tree. It's consistent with this thing called the eocyte tree, which was proposed originally by this guy Jim Lake at UCLA. Here are the trees, and then I have a zoom in on the trees. So what they do first, this is the ribosomal RNA data. They compare in A and B what happens when you add the new taxa versus don't include the new taxa. And this is their proof that taxon sampling is really important. So if you ignore A first and you look at, this is basically what you would have expected prior to their study. They redo it. And what they find is that when they don't add new taxa, they get the 3-domain tree. They get bacteria here, archaea as a monophyletic single, all the archaea grouped together with a common ancestor here, and eukaryotes as a separate group. And then if you add new data representing more diversity of archaea, you see this, which is that there's one group of archaea over here, another group of archaea over here, and eukaryotes are embedded in the middle of the archaea. They are not their own separate branch. So that their argument is basically this tree on the left with the new data does not support the 3-domain tree of life. Does that make sense?
Have you done more work on this since? This was
Oh, that might be your homework assignment. It may have been a paper that came out a couple weeks ago on more work on this. So then they go even further. They say with the ribosomal RNA data, what if we actually really fine-tune it and use preferred models? So when you take sequence alignment data and you calculate a tree, in most cases you have to have some model of how sequences change over time in order to calculate which tree is best. They say, what if we use better and better models? What does that do to our tree? And they find that the eukaryotes are still embedded within the archaea when you use better models. And in fact, with what they think is their best model, they're actually 1, 2, 3, 4 different archaeal groups, and eukaryotes as just sort of one branch of the archaea. And then they say they did not, resolve the 3-domain tree, which they said was surprising. They're lying there now, by the way. They have not believed in the 3-domain tree for 20 years, but they can say whatever they want. I'm going to give Martin Embley shit about this. They then asked, what happens if we carefully or don't carefully select which data we use from our alignment? This is that thing I showed you called masking. You take a sequence alignment and you say some of the data is good, some of the data is bad. They think that when they built their tree, They were pretty conservative. That is, they used data that was only really, really good. But what happens when you use different portions of the data and they get the same result? So it doesn't appear to be, for the ribosome RNA, dependent on their stringent masking of the sequence data. And then they say this, increasing taxon sampling is great, but we don't have enough characters, or maybe we don't have enough characters. So now it's the same argument as in the Laura Katz paper. We've increased taxon sampling. Let's also increase character data sampling. So now they go back to these same organisms, and they scan through the genomes of these organisms for sets of genes that they can identify as homologous between all of them. They identify these 29 sets of genes, and that are across all bacteria, archaea, and eukaryotes, and 64 genes that are across all archaea and eukaryotes. And then they do phylogenetic analyses of these. Same issue. What are we going to do if there's horizontal gene transfer? Let's compare the topologies of these genes to each other and identify the ones that agree with each other. Which ones are congruent with each other? They actually use the same software that the Laura Katz group used. I've never heard of this before reading these papers, but they use the same software to find the congruence. They identify sets of genes that appear from within their data set. So within their 29 BAE data set, 29 proteins across bacteria, archaea, and eukaryotes, they identify subsets within there. Set of 10 of the genes and then 6 of the genes. These 10 genes seem to have the same phylogeny. Let's build a tree with those. These 6 genes seem to have the same phylogeny. Let's build a tree with those. So they build trees for each of these sets of genes that were inferred to be congruent. And this is the result with their protein coding genes. And again, eukaryotes are a subgroup within the archaea. So, basically with ribosome RNA, with concatenated ribosome RNA, now with diverse protein coding sets, they find they cannot find 3 domains. And they find more groups, especially that the eukaryotes are just a subgroup within the archaea. And they focus on this TAC superphylum that they mention in the introduction. It basically comes from the names of some of these individual archaeal lineages. Thaumarchaeota, I don't even remember what this one is, Corarchaeota and Crenarchaeota, this TACK group, with so-called better sampling. This goes to your question. This is actually, horrible sampling still of bacterial and archaeal diversity, which might come up in the new paper. But it's still better than what was done previously. And then they go through a lot of different things that they use to test the statistical support they saw for the particular patterns in these results. And they also went through whether or not the thing that they were really interested in next. So now they've convinced themselves, although again, they convinced themselves 20 years ago, that the 3-domain might be wrong, and there might be this one, eukaryotes might evolve from within the archaea. And now what they really wanted to know is which archaeal group is the sister group to eukaryotes. Of all the archaea, is there one lineage that is most closely related to the eukaryotes? And this would be the dream of everybody who studies archaea could now apply to NIH and say, hey, I'm working on the sister group to eukaryotes. That's relevant to all of human medicine and cancer and neurobiology, and therefore you should give me money because this archaeal group, that is basically what they're looking for here. And the archaea are generally pretty poorly studied. So it may be that there's a lineage out there of archaea that we know virtually nothing about that could have come from just random genome data, and we might not know anything about its molecular biology. And if it is the sister taxa to the eukaryotes, that might be useful to study in terms of learning about the evolution of the nucleus, the evolution of all these eukaryotic features. And we didn't talk about this, but whatever tree you believe, in most studies it's pretty clear that eukaryotes are monophyletic with each other compared to all the other lineages. And a ton of interesting events have happened in the evolutionary history since the eukaryotic lineage separated from the rest of the gamish of the well-studied bacteria and archaea. And we are very interested generally in those features like chromatin and introns, uncoupled transcription and translation, the presence of a nucleus, the use of actin and tubulin for the cytoskeleton, and all these diverse features of the eukaryotic cell. Maybe there are some archaea that would be better models for studying the evolutionary history of that than others. And that's what they spend the next portion of their paper trying to figure out or test for. Is there one of these archaeal lineages that we can figure out is most closely related to the eukaryotes? Now, unfortunately, when they did this paper, there was not a good syncing with this paper. So by not having a good sampling of eukaryotic diversity in their study, it limited their ability to detect which archaeal group was the sister to eukaryotes due to long branch attraction and due to taxon sampling issues. But they try. So they go through their data and they basically try to figure out which one it is. And they say, with the exception of this one core archaeal group, they couldn't find any statistical support for any of the archaeal groups being the sister group to eukaryotes in this study. There's a little bit, like a smidgen of a hint of some connection to this core archaeal group. They then, this is pretty controversial, right? I mean, 3-domain is in most textbooks that are modern, and before that it's the 5 kingdoms, so there's no textbooks that talk about the eukaryotes being a subgroup of archaea. So they spend a lot of time in their paper testing the evolutionary models that they're using, testing the gene set, the effect of the gene sets on the result that they get, and a variety of other sort of sampling issues. They even developed this new method to calculate congruence among trees. And what they do is they basically, there are many methods for doing this, but let's say you have a tree that relates these 4 species, and you have another tree that relates those same species, but it's different. There are many methods that are available for saying how similar is this tree to that tree. And they developed yet another metric for doing this. I actually don't like that they did this because this metric is new and not very well tested, and we don't have a literature to say what it means. But they make the argument that you need a metric that does things like incorporates information about the length of the branches, not just the topology here. And then they show you some information in the last figure about the data sets that they're looking at. So the one that's I think is most useful is this part A, which is basically for their data sets, they take all the trees that they get and compare them all to each other. And they ask, how different are they from each other? And they see this bimodal distribution, which is a little bit not what they expected probably. So the bimodal distribution could be that there are 2 sets of evolutionary histories in their data that are very different from each other. And that, evolutionary history 1, many genes show evolutionary history 1, and the trees are pretty similar to each other, so they have a low distance from each other, but they're really distant from the other tree. And then the other genes show the other tree, and they're all really similar to each other and not to the other tree. And you'd get a bimodal distribution. They actually find this really interesting thing, which is that all of these large distances, they argue, are due to the one taxon being misleading in their data set. I think it's one taxon, not one gene. And so they then argue that on average, the genes that they're looking at do in fact have roughly one history behind them. And you get a normal distribution for the rest of the data. And when you have one history, you can then sum up across many of them to get the average across them. So this is basically standard operating procedure for most deep evolution papers now is you would never get past review at any journal if you didn't do a lot of these tests. You can't just take data, build a tree, and say, oh my God, my tree is different than everybody else's, here's the result. They probably spent 2 years on these statistical tests since they had that original result. And then they basically just talk about how with this new data set, they find that the eukaryotes are a subgroup of archaea, and then, just as a throwaway at the end, totally unrelated to anything else they're doing, they say, interestingly, members of this TAC group, so if I go back to the tree, this TAC group, which is half of archaeal diversity, all the eukaryotes are always more closely related to at least something in this group than they are to this second group of archaea, the Euryarchaeota. So they focus on this TAC group, even though they can't figure out which member of this TAC group is closest to eukaryotes. The whole group, something in it is closest. And they say, interestingly, this TAC supergroup, many of them actually encode interesting genes that previously were thought to be eukaryotic specific. Cell division machinery, the ubiquitin protein modification system, actin, tubulin, and other features. So that's like their little throwaway at the end that, it's going to be important to study this TAC group for model studies of eukaryotes. You should give us money to do our further experimental studies. So are there, so that's basically, this is the modern view of the tree of life until you get to some paper you might see in the next day or two. And that, it's pretty different than the WOS 3-domain concept. But in fact, as they showed in this figure, it's not that anybody made any mistakes previously. With the best methods available, they still get the 3-domain tree. It's just that 3-domain tree, they basically argue, is a mistake that is based upon poor taxon sampling, probably due to this long branch attraction issue, where these two archaeal groups that were pulled together in this tree really should be split apart. And when you sample the taxa better, they get split apart. And they don't even, basically say these novel groups that were discovered as being separate from bacteria, they're still interesting. They just say the eukaryotes may not be a completely separate lineage from the archaea.
Quick question.
Yeah.
Could you imagine a wider screen that would had even more new data? And then
Well,
get
so
another
yeah,
organization?
you'll see that in the next
Oh.
paper you will see. That was one of the reasons why we did this project that I coordinated called the Genomic Encyclopedia of Bacteria and Archaea, where we marched our way through the evolutionary diversity and gathered genome data from across the diversity of bacteria and archaea. And the main problem with that, as we will see when we get to the core next week, is that most of the diversity of bacteria and archaea is represented in uncultured organisms. And all of this data is from cultured organisms. And if you really want to resolve the phylogeny, we need to sample the true diversity of eukaryotes, bacteria, and archaea. And we've also done that, or a woman at the Joint Genome Institute, Tanya Woike, coordinated a project to sample uncultured bacterial and archaeal diversity. And we wrote a paper on that we will talk about later in the course. And even with that, even with 300 new genomes that she sequenced, we still think we have maybe 1% now of the total diversity. So it's still, I think that this pattern could easily change with more sampling.
So
Yeah.
that would fall under metagenomics and stuff.
So the new paper was actually done with single cell genome amplification. So it's sort of like culturing, but your culture is one cell. We didn't do random sequencing from the environment, although people are also doing that to try and get. The problem with random sequencing is it's hard to build a whole genome. And if you want to concatenate sequences of different genes to increase your signal, you need to know that the different genes all came from the same cell. And so we needed single cell genomes to do that. But we will read that paper. And the person who led that project, Tanya Woike, is giving a seminar here in a month. I'm going to send the announcement around. She's completely, amazingly brilliant. And she and I are working on a grant proposal to fill in the rest of the tree.